This is a brief review with cartoons and animations on how cytokines signal through the Janus kinase activation. So in this cartoon, basically what I'm showing here is a brief summary on the more well characterized or better characterized TH profiles or phenotypes that have been associated with diseases. Starting with TH1, this particular phenotype is very well characterized as an inflammatory response. It is a response to danger, and basically it's the response we typically will have when we get a sunburn or a cut, or we are dealing with most pathogens, bacteria, etc. So this is a profile that's characterized by the phenotypic specialization of the T helper cells into the Th1, and it's going to cause a very inflammatory response. The Th2 is a response that's very characteristic of allergies, allergic responses, and this is the way that the body has to deal with different kinds of allergens, anywhere from food allergies to eczema to mosquito bites, and of course the diseases are very allergic. The Th17 profile is associated with the Th22 profile in humans, and basically the diseases have a component of inflammation, but they have been very well characterized. And it's typical, very actively typical for psoriasis, intestinal bowel disease or inflammatory bowel disease that comprises Crohn's disease and ulcerative colitis, and rheumatoid arthritis, psoriatic arthritis, ankylosing spondylitis. So these particular phenotypes are characterized for the TH response. And finally, we lead with the tolerance or immunosuppressive profile of the T regulatory cells FOXP3. These are not linear. As I'm showing them here, they would be seemingly independent. But now we know that there's a lot of crosstalk and a lot of control and communication between these different phenotypic expressions of the different TH cells. And again, they can occur either in the normal situation dealing with danger, or they can be associated with very specific diseases. So the Th1, one of the products of that Th1 differentiation is interferon gamma, which was also one of the triggers that made the pluripotent T cell differentiate into Th1. So there's a bit of a retroactive loop that's positive, building up on that response. And this is something that we have all lived through when we fall and have a trauma, when we cut ourselves, or dealing with diseases. In a first stage, there's an increase. There has to be an augmentation of that particular cell that is perfect for dealing with that particular response. In a second stage, the products of these Th1 can also stimulate some of the cells that are characteristics of the Th2. In this case, particularly TNF has been shown to stimulate the B cells, which are very typically a response of the Th2. Now, interleukin-2 is inhibiting the differentiation to Th17, and it's a major product of the Th1. But it also inhibits the B cell, the isotype switch. So in other words, you have TNF stimulating the B cells, but IL-2 from the Th1 profile inhibits the switch. So the most characteristic B cell response in Th1 inflammatory responses is not going to be switched. It's going to be either an IgM, like when we have the rheumatoid factors, or it can go to IgG, but you don't see the full switch of the B cell a heavy chain to an IgE, which is typical of the Th2 response. So in this sense, there's a combination of crosstalk and there is a specialization into what is going to happen for that particular profile. The Th2 profile also has this sort of retroactive loop that's providing a positive feedback into producing more Th2s. And of course, with all the allergies, we do know that, you start sneezing, you start itching, and originally there's a very fast increase in that particular Th2 response. The Th2s will produce interleukin-4, which were also the cytokine that differentiated the pluripotent T cells into Th2s. But also some of the products of the Paneth cells and epithelial cells that are the distal end organ target of the Th2 response will be producing some cytokines that will stimulate further Th2 development. So there's this retroactive loop of growth. And there's also this crosstalk. The B cell is actually not only responding to interleukin 6, but it's also producing interleukin 6. And interleukin 6 can actually have signaling effects on the inflammatory pathways, not only, as you can see here in this cartoon, having effect on synoviocytes, fibroblasts, and even osteoclasts and cartilage. But if you go left in this cartoon, interleukin 6 also has effects on the differentiation of Th17s, Th22s, and Tregs. So the production of some of these interleukin 6 by the stimulated B cells can actually lead to also some stimulation of other pathways in this Th phenotypic differentiation. Likewise, as all the other pathways, Th17s will produce interleukin 21, which is one of the cytokines that actually produces differentiation from the pluripotent cell into a Th17 cell. And interleukin 17, which is the major product of the Th17 profile, will inhibit the differentiation into Th1. So you can see right now here that generally a differentiation into Th1 inhibits via IL-2 a differentiation to Th17, and Th17 differentiation via directly interleukin 17 inhibits the differentiation into Th1. So there's a little bit of a control mechanism here that would probably tell us that diseases will have either one or another phenotypic profile, which would be more typical of a particular disease stage or even individuals. So not all diseases are going to be the same during the disease progress and also in different patients. Interleukin 17 also inhibits directly NK cells, which are a major target of Th1 responses. Now, what we see then is that almost all of the products of all of the other differentiations, either peripherally from neutrophils or even from the first Th phenotypic responses, the production of TNF can come from many different places. Interleukin 6, as I mentioned, also can be produced in different stages and different phenotypic expressions, and interleukin 2, together with interleukin 27, will eventually lead to a tolerance or immunosuppressive profile typical of the T regulatory cell. So all of these is actually characterized, but the important factor for this particular image is to say, how do we know it's a Th1 or a Th17? It's the cytokine profile. So the cytokine profile not only drives the differentiation on the very left, the first column of interleukins listed in there are cytokines. Not only the cytokine, the specific cytokine profile, it will determine what is going to be the differentiation, either Th1, Th2, Th17, 22, or Treg, but also each one of these will be characterized by the secretion and production of a very specific cytokine profile that helps identify what is the response we are seeing. So that's why it is very important to understand cytokine pathways and Th phenotypic profiles in the concept of the diseases that they're associated with, but also how they deal with danger. Because when trying to modulate these, if we understand what they're there for, we have to remember the immune system is not there for to cause diseases. It's there to deal with danger. So if a particular disease has a predominant pathway, we need to also remember what is that pathway normally used for and what could be the potential adverse event profile expected if I modulate that pathway. So the binding of cytokines or the engagement of homing or co-stimulatory factors will produce an internal signaling, and blocking that particular interaction extracellularly has been well received and well characterized using biologics. But there's another way that we can also block that signaling. So what happens when the cytokine or the co-stimulatory factor bind the receptor on the cell through the extracellular domain? This actually brings an activation basically by phosphorylating the kinases and the different secondary messengers inside the cytoplasm and eventually produce gene transcription. So, this could be just a brief example, could be IL-23 is binding the receptor, activates the kinases, and the gene transcription is producing interleukin 17. So this is just one example how interleukin 23 actually is differentiating that T cell into a Th17 cell that is secreting IL-17. So we can block that with external blockade by either small molecules or large biologics, or we can find the alternative of small molecules that can target these intracellular pathways. So in this case, the small molecules penetrate the membrane. It's not like the large biologics that they have to stay outside the cell. The small molecules penetrate through the cellular membrane into the cytoplasm, and they actually bind any one of these particular areas where normally the kinases would be phosphorylated. But by not being phosphorylated, by blocking the phosphorylation, note that the engagement of the extracellular ligand can still happen. But basically what you have is a kinase that is not active, and by not being active there is no gene transcription. So either blocking extracellularly and not allowing that binding to happen, or allowing the binding to happen but not allowing the activation intracellularly to happen, we have the same result of no gene transcription. So bottom line, that cell will not be responding. And that's another way of blocking these cellular crosstalk and communication. So there are several receptor superfamilies, and what I put in here was just reminding you that these cellular receptors are present in many cells. They're not unique to the immune system cells. They're present in many cells, and that's what allows cells to produce the cross-talking, the communication, and the responses. The receptor superfamilies are characterized by basically internally. So what I depicted here in blue is like the cytoplasm, and then you of course have the nucleus and the gene transcription in the bottom. So the first superfamily is a type 1-2 of cytokine receptors, and they are characterized by having tyrosine kinase association, and they signal via the STATs, the signal transduction and activators of transcription. So this is the way that the first family will be communicating to the gene. The second superfamily is the one we know as the toll-like receptors, which is the one that the interleukin-1 uses. And the intracellular signaling is a bit more complex. It involves MyD88, which is an adapter protein that will produce several intracellular messengers for the signal activation, which is TRAF and IRAK signals, and then they go to the NF-kappa B for gene transcription. So signaling internally, the first one is tyrosine kinase and STATs, and the second one includes TRAF, IRAK, and NF-kappa B. The third family is the family that's the NF-kappa B family and TNF. This is the one that tumor necrosis factor uses. And also the BLIS for the B-cell activation uses this kind of receptors. Basically, this is trimeric receptor, so there's 3 chains, and they will signal via the NF-kappa B and the MAP kinase. We have the TGF-beta receptor. They use serine or threonine kinase, as opposed to the first type 1-2 cytokine, which used tyrosine kinase. The TGF-beta receptors use serine or threonine. So that's what differentiates this superfamily from the others. The chemokine family will now use CAMP, so cyclic AMP. It's a G protein. It's not the very typical of the others. This is unique for the chemokine receptor, and they also be activating via the phosphodiesterase 4. So the PDE4 is the via of communication for intracellular signaling. And finally, the last of the 6 superfamilies is the receptor tyrosine kinase. So in this case, the enzyme-linked receptor. So this particular receptor does have the tyrosine residues within the chain, as opposed to the first one, which doesn't have a kinase in the receptor, but the kinase is associated physically. It's a different protein associated to the receptor in the type 1-2, whereas in the receptor tyrosine kinase, the tyrosine residues are in that receptor. And that's what characterizes the 6 different families of receptors. It is important to have an idea of what exactly is the mechanism for a particular communication or pathway in disease stages, so that we can understand what exactly are we modulating, where are we modulating, and what can be the expected results of this modulation with the different armamentarium of drugs that we have. In this particular cartoon, again, this is a very complex and hypothetical cell that is showing all the different crosstalk of the different receptors, but in the JAK system, the JAK signaling cascade, it's a very direct cascade via the receptor, the Janus kinases, and the STATs, the signal transduction and activators of signal translation and activator of transduction. So the way that the JAK or Janus kinase system works is very interesting, and there's a beauty in the simplicity of it. There are 4 JAK family members. it's a JAK1, JAK2, JAK3, and TYK2. There are 7 signal transduction and activators of translation, which are STAT1, STAT2, STAT3, STAT4, STAT5A, STAT5B, and STAT6. The Janus kinases are associated with the internal portion of the receptors. So these receptors, which are the cytokine type 1-2 receptors, in the intracellular tail will have 2 Janus kinases associated with them. And these 2 Janus kinases that associate with them will depend on the receptor. As you can see in this cartoon, we have different cytokines, and this is just an example. So the gamma chain cytokines are the cytokines associated with the white blood cell lineage. And then you have the interferon that will bind a receptor that has a different pair of JAKs associated. Interleukin 10 and 22, interleukin 23 and 12 have TYK2 and JAK2, and so on. So different cytokines will have slightly different specialization for the cytokine that they're binding, and intracellularly the pair of JAKs that they have. Just note that the IL-6 and IL-11 column, the intracellular Janus kinases are 2 of them. But for this particular group of cytokines, any combination of these 3, so it could be JAK1 and TYK2, JAK1 and JAK2, JAK2 and TYK2, any one combination of a pair with these 3 kinases will be associated with the receptors of the cytokines, IL-6, IL-11 families, that will signal through the STAT1, 3, and 5 for transcription. The only example that I know of, and to date I don't think there's been a publication on any other example, where 2 of the same Janus kinases are associated in a receptor, is a typical example for EPO and for the CSF growth factors. These are not cytokines, these are growth factors. And that's very important for the cell to differentiate which kind of stats and which kind of signal you're going to give to the cell. In the other signaling, what you could be signaling is a synthesis of a different cytokine. In the EPO and the growth factors, what you are signaling to the cell is basically replication of that particular lineage cell. And this is a way they have to communicate. So it's very important to understand that the 4 JAK family members have different combinatorial abilities, and also via 7 different STAT members on different cells, we can have a tremendous amount of different kinds of responses. So how is it that only 4 Janus kinases and only 7 signal transduction and activators of translation can actually produce so many different results? Well, I'm no mathematician. There's a mathematical field on combinatorial power and the power of combinations. This is just a schematic that can actually show how having 4 elements, you can have how many different combinations, and of course then you have rules of combinations. You can say X does not combine with Z, but X can combine with Y, whatever it is. So these combinations actually create a much richer amount of data than if we had to have each one as a whole. So power combinations is a complex field of mathematics that we actually use every single day of our lives, because we all use 26 letters that have the power of being combined into words. And just a few, a little question for you if you're listening and paying attention and liking this. Just have a wild guess. How many words can you create with 26 letters? And of course, there are some rules of combinations. Uh, Sometimes the rules can be X letter cannot go before the other one, or you cannot end a word with a particular letter, but in general, you actually have a power of combination that is tremendous. So if you guessed around, I don't know, 200,000, you probably were on the right track. But that's only if we're counting English. These letters can be combined to form words in many, many languages. So the power of combination is tremendous, and in the cell, having the minimum amount of elements allows the cell to have less DNA committed to having a message. So if we had to have one gene for each one of the proteins, or one amino acid, one nucleic acid for each amino acid or for each protein, I think we could not be an average weight of 60 kilograms or 120 pounds. We would probably be 1,000 times more heavy, or even more. We could not contain all that amount of DNA in our cells. So we need to be economical in the cell and make sure that we have the minimum amount of elements with the maximum combinatorial power, so that all of these different combinations give us all the different responses that we need to have in the case of the immune system, to deal with all of the different perceptions of danger that can be from pathogens to sunburns or cuts, and also they're used, unfortunately, they can produce diseases. But this is the important thing about combination and about signaling of cytokines that, for instance, are using the Janus kinase system with only 4 Janus kinases in combination. So when we look at the 4 different JAKs and the different receptors that have been described, we have the JAK1, JAK3 receptor, which is the receptor called for the gamma chain cytokines, the JAK1, JAK2 receptors associated with that pair of JAKs. We have the receptor associated for the TYK2, JAK1, the TYK2, JAK2, and then the 2 combinations of any one of these 3. TYK2, JAK2, JAK2, JAK1, JAK1, TYK2, etc. So these combinations also, in association with different intracellular signaling of the stats, is going to be very important for the signal. So the gamma chain cytokines are basically associated with the white blood cell lineage, and they will be present in basically the cells of the immune system. The JAK1, JAK2 are more commonly used by interferons and basically produce a STAT1. This pathway is a pathway that deals a lot with viral diseases. The interleukin 10 and interleukin 22 are going to use different pairs of JAKs and different stats, the 1, 3, and 5. And interleukin 10 is a critical cytokine for regulation and immunosuppression or tolerance. So using the IL-10 in a very specific way with a specific stats, it depends on the cells that are expressing it, depends on the cells that are producing cytokine, and it depends on the stats that are going to be activated, what response we have. Is it an IL-22 response or is it an IL-10 response? In the case of IL-23 and IL-12, it's also very important to know which is the cell that is actually responding. IL-12 will be basically signaling through the TYK2, JAK2, and activation of the appropriate stats to produce a Th1 phenotype, whereas the IL-23, using the same TYK2, JAK2, and different stat pathways in different cells, will be producing or differentiating that Th phenotype into a Th17. So we have the same receptor, the same intracellular JAK association with different stats in different cells, two very different phenotypic expressions of the Th, either Th1 phenotype, more inflammatory, or the Th17 phenotype. And then in the case of the IL-6 and interleukin-11 and others, we have more combinatorial power, and we have also different stats intracellularly. So these combinations allow for a lot of different responses. IL-6 is actually present, and it's activating, and it will bind many, many different kinds of cells. It will produce many different kinds of responses, basically many of them inflammatory, and it will also be involved in triggering IL-10, which is a regulatory tolerance mechanism. So IL-6 has a lot of different effects in a lot of different cells, and all of these different ways of communicating with the cells has to do with this combinatorial power of the JAKs and the STATs. And finally, the growth factors. So epoetin, thrombopoietin, and the granulocyte colony stimulating factors, they basically will be signaling through the only known, at least to me, I don't think there's been another identical pair of JAKs identified for other signaling, but at least the pair of two identical JAK2 associated to these receptors are unique for growth factors. These are not for cytokines. And the stimulation via the STAT3 and 5 of these particular receptors and this association will produce cell growth. So these are growth factors for the epoetin, thrombopoietin, and neutrophils. So when we have cytokine signaling via the JAK family, that it is simple internally, but in this case, it's a very complex externally. So let me explain why I say that. We have the 4 different JAKs in this table, in the columns, but what you can see here is that internally, we can have different pairs of combinations of the JAKs that signal through particular stats. And that's what you see there. Stat 5 uses IL-7 for JAK3 and JAK1, IL-9, IL-15, IL-2, IL-4, and IL-21. These are the gamma cytokines, and they are present in the white blood cell lineage, and they are associated with an adaptive immunity response as shown on the top right. So JAK1, JAK3, many different cytokines. Intracellularly, more simple. Since these two have to be associated and active, just inhibiting one of them intracellularly can inhibit the signaling of all these kinases with only one single intracellular inhibitor. If we move on to the JAK2 TYK2, you see the IL-23 and the IL-12 will actually be binding a receptor that signals through the JAK2 TYK2. So again, these particular cells will be responding to the different cytokines, the 2 different cytokines, and you can inhibit that by having only one of the 2 JAKs inhibited. Now, if you go down the list, for instance, and you see, what if I were to inhibit only JAK1? You look down the list and you see all of the cytokines going all the way that have not only the adaptive immunity but inflammatory T cell differentiation, innate immunity, they all would inhibit a lot of different cytokines. If we had to inhibit these externally, extracellularly, we would need to have, uh, well, probably an antibody for maybe each one of them. Maybe some of them can share some elements. But in general, to inhibit all of these cytokines, you can do only with one single targeted intracellular kinase, like the JAK1. That's one example. If you inhibited the JAK2 only, you are going to also hit the growth and hematopoiesis, um, the signaling for the growth factors. So in general, for treating inflammatory diseases, ideally you want something that does not inhibit JAK2 or that inhibits JAK2 at different concentrations or has lower affinity. And at the dose that you will give it to your patients, you're expecting it to block the other JAKs and not the JAK2. So, in this cartoon is just a reference for saying intracellular inhibition by using the system, the cellular system of the power of combinations that has less amount of individual proteins working to produce many different signals. You can target one of those more simple intracellular mechanisms and block many different cytokine signals that could be involved in disease.
Thank you for listening. I hope this was useful, and I've put in here the key references that I have used to create the many different cartoons and animations. I will also post this in the YouTube description, and please come back and listen to this again. Sometimes it's necessary to listen to this several times, and feel free to check other YouTube videos that I have posted, and also evidently look at many other references.